first one we'll talk about, I'll have a slide in each of these, is something called gap junctions, which is a physical connection between two adjacent cells. We'll also look at something called contact signaling, which is where two cells side by side touch each other and communicate that way. We'll also look at some very short distance chemical messaging, the autocrine and paracrine signals, and then we'll finally look at a couple of modes of more longer distance regulation. So each of these are going to be outlined on one or two slides over the next few minutes. So let's start with the first one, gap junctions. Gap junctions is technically pretty easy to think about. It's imagine two different cells where their attachment has a pore that allows for the movement of things from one cell to the next. So if this were a room, if this room were a cell, and the next door room was a cell, we can open a channel and we can allow things back and forth. That's essentially what gap junctions are. So the gap junctions are formed by these specialized proteins. I put the important terms in red in case you want to try to remember a few of these. Essentially, we have these things called connexins or connexons, which are these little connections that essentially fuse these two cells together. And the cytoplasm of each is literally linked. So if we look at an actual surface electron micrograph of a heart cell, all of these little dots are actually these little gap junctions. This cartoon is trying to show you a little bit more complicated view of what we have here. Essentially, these tubes or channels, they can allow certain things across, allowing those two cells to literally send messages back and forth. The channels can be open or closed, so the cell can regulate that, and therefore things can be passed. The most common cell type we have that utilizes this mechanism is our heart. Our heart cells have gap junctions, and this is how our heart cells tell each other that they're about to contract. And so the contraction of your heart is regulated through these gap junctions. Essentially, by allowing charged ions, charged ions like sodium and potassium, to transfer back and forth from one cell to the next. Okay. So a very simple but effective way of communicating. Not very common in the animal kingdom, but in cases where they are used, they're very, very important. Okay. So that's our first of a handful of communication methods or techniques, the gap junction. The next one is called contact-dependent signaling, and this is exactly what it sounds like. It's the surface of two cells in contact with each other, literally sending messages back and forth. So imagine you elbow your person next to you to pay attention in lecture or whatever you're trying to talk about. You're communicating through an external sort of force. You can't do that with the person on the other end of the room because you can't reach them. Some cells have these extracellular attachments to one another that can be used to push and pull on cells next door to send messages. So like when you eat, your stomach cells will stretch, and they'll stretch and pull on one another, creating signals which can lead to things like satiation and other things. So there's actually quite a few times where this kind of mechanism can be used to tell each other what's going on. So again, these are on the surface of the cell. Surface molecules bind one cell to the next. And if you want to get into it, there's these really complicated structural elements that can link cells together. What's really fun is that the name for these surface molecules that attach the cells together was originally called CAMs, or contact adhesion molecules, because they were first discovered and thought to be important just for holding cells together. Very basic, how do cells hold each other together? But now we know these CAMs can have much more complicated roles, as in sending messages. So essentially, something in one cell can change in shape. This is the skeleton of the cell, the cytoskeleton. Pushing and pulling on this extracellular part, they can then push and pull on its neighboring cell. So even though we thought these were pretty basic, they actually have a lot of roles to play in sending a message from one cell to the next. So these are called contact-dependent signaling, or CAMs. Again, very simple, only works from one cell to its neighbor, kind of like the gap junctions, just a different mechanism by which they work. The next process is a little bit more complicated. It's the chemical signaling that is most common in the body. This is collectively either known as autocrine or paracrine signaling. I'll tell you the difference between those two terms in a second. So imagine you have a cell here that's able to release a chemical into the extracellular space. So that little red diamond is being released. That little red diamond can now diffuse from wherever it's released to nearby areas. So it's a very simple process, release a chemical and let it drift around. But that chemical may have a receptor somewhere else that can attach to it and essentially get a message. Okay. So those chemicals are generally called ligands or ligands, and a ligand or ligand is just a message. They come in all kinds of different flavors and colors and shapes, depending on whatever it is. If another cell, or as you can see in that first cell, its own cell has a receptor for it, it can elicit some kind of response. So autocrine signaling is when one cell releases one of these ligands or chemicals, and then that same chemical binds to a receptor on itself. Okay. So essentially it's the cell telling itself something, maybe a different part of the cell further along. So that's what we call autocrine. That's what's shown in the first slide, or first cell. But if that ligand is released and maybe drifts to the next cell or a few cells away, same message, same reaction, it's called a paracrine signal. So the same kind of idea, it's just whether or not it is responding from its own self or to a different cell. So that's the only difference between these two words. One example of this is if you get scratched by a cat, and if you have a cat, you know exactly how that feels. If you have a friend with a cat, you probably know how that feels too. I don't know anybody that has not been scratched by a cat. I don't have a cat for this reason. So the cat scratch is going to damage tissue, creating obviously the wound. The damaged tissue will release chemicals, and they will drift beyond and away from that point of damage. One of the chemicals that's commonly released by wounded cells is histamine. Histamine will bind to receptors in other cells, creating a response, essentially an inflammatory response. And that response is important for wound healing. So it'll lead to more immune cells coming into the area, more blood flow to the area, more swelling, which will help with the wound healing. So a little chemical released from one cell affects all the cells nearby in this paracrine fashion. Again, we're talking about communicating between cells at a very short distance, but it's still really effective. Once we get to much longer distances, though, new and more advanced ways of communicating had to evolve. And so we're going to talk about a couple of those, a couple of main areas of long distance communications, where messages are going to be sent from one place to the other. The first one is the endocrine system, and I'm sure you're all aware of what that is. This is how our hormones are used to send messages from place to place. An endocrine cell, or an endocrine gland, which is made up of endocrine cells, is going to release some form of chemical, which is then going to enter the bloodstream and travel to potentially far off places in the body, where they'll come in contact with other cells, and any cell with a receptor for that chemical might respond in a certain way. So the endocrine system uses these chemicals, and we call those chemicals hormones when they're in the endocrine system. So this is potentially useful for sending messages wherever your blood goes, which could be a very long distance in the body. But of course, the blood only travels so fast, so it can only go so quickly. So maybe many seconds to minutes or hours it might take for a message to go from one place to another and elicit some kind of response. The endocrine system is obviously a really important way that we communicate throughout our body, and we see it evolve into pretty advanced form in many animals. Very similar to the endocrine system is something called the exocrine system. And exocrines are similar to endocrines in that they're a chemical released from a cell that travels somewhere else to have a response. The only difference is the way that the chemical gets from point A to point B. So in an exocrine system, there's a physical duct or path or route by which the chemical goes from its point of production to find its receptor. So it doesn't use the blood to get from place to place. It uses a direct ductule or pathway. So one great example of that is the pancreatic duct. Cells of the pancreas will produce all kinds of different chemicals that will be released. They will travel down the pancreatic duct, and the pancreatic duct ends at the small intestine. And so these chemicals are now released into the small intestine, where cells will bind them, and then all kinds of reactions may occur. So the pancreas is considered an exocrine gland. The pancreatic duct being that direct connection between the gland itself and the cells that are going to respond to those chemicals. And then the chemicals are given the word or name exocrines instead of endocrines or hormones. So they have different names, but they're doing the same thing. They're just chemicals that are released, travel somewhere else, and bind with a receptor. So we have a bunch of these different glands that can do this type of thing. Again, not using the blood, but using the ductule as their point of, or their method of movement. Any questions so far? I've kind of like given you a whole bunch of information all at once, just to whet your appetite on communication. The next form of long distance communication, I'm sure most of you are excited about, because a lot of you are neuro students, is obviously the nervous system. The nervous system is very advanced and very quick to send messages from place to place in the body. And what it essentially does is it combines two things. An electrical signal that can be passed very rapidly from one place to another in the body, and then a chemical signal that can then lead to some kind of response on a target. So a neuron is the main cell type in the nervous system that does this. It can send electrical signals along its length. And the length of these cells could be many meters or feet in length, depending on the type of the animal. So you have neurons that go from your lower back all the way down to your foot. One cell stretching one long distance. And they can send an electrical signal at about 100 meters per second. So this is one of the fastest known mechanisms in the animal kingdom. It's up there with those cnidocytes that the jellyfish use. Very rapid electrical signal being sent from one place to another. Imagine flipping on the light switch, electrical conduction going along the wire, and then when it gets to the end, the cell releases a chemical. And then the chemical moves to a nearby cell and binds to a receptor. That's how the nervous system works. And you can see it's a connection between electrical signaling and chemical signaling. The chemicals involved in the nervous system have their own name, neurotransmitters, but they're working just like the endocrine system and the exocrine system. It's a chemical that's released that binds to a receptor. So this is a very advanced and effective form of communication. Potentially from between long cells that are very far apart, they can act very, very rapidly. So as we see animals evolve into more complex structures and forms, we're going to see that the nervous system also evolve into far more complex systems too. Now there is one more that's sort of a blend of the nervous system and the endocrine system. So I'm going to introduce that to you in case you come across it. Here's a situation where a neuron, so this is the nervous system, releases its chemical into the blood. And then it travels through the blood to its target. So this is kind of a blend of the nervous system and the endocrine system. And so for that reason, the chemicals are called neurohormones. Okay, so everything has its own name. But that's really all the differences. There's still chemicals get released that find receptors in different places. Okay, so with a suite of different mechanisms at our disposal, we can find ways to communicate from one cell to the other, keeping our whole body in tune with what's going on in other parts, and then regulating and reacting to things as we go. Any questions about these general ideas as we wrap up this section? So generally speaking, we have a signaling molecule that's released by a cell, and it's going to bind to some sort of receptor. Okay? It could be next door, it could be on its own cell, it could be very far away. We're not going to get into this today, but if you take cell biology, you'll see that there's a whole suite of different signaling mechanisms and different reactions that cells may have to this binding of this chemical, leading to some form of response. This is the general pathway by which it works. But what I do want to point out is that the receptor, even though all the images I've shown up to this point were always on the cell surface, sometimes they're actually inside the cells too. So a lot of times we're on the cell surface here, little receptor for chemical, but sometimes we actually see the receptors right inside the cell, in the cytosol, cytoplasm. And in other cases, it may be right inside of the DNA in the nucleus. So a signaling message, if it can get into the cell, might elicit a response right at the level of, DNA creating new DNA or cell division or all kinds of different things that might happen, proteins leading to protein synthesis or other things. So the way that it happens can be varied, but the general idea is the same. Chemical binds to a receptor somewhere and elicits some kind of response. So keep those things in mind as we advance into much more complicated organisms. When you take other classes like physiology, cell physiology, or cell biology, any of the neuro courses, you're going to talk about these types of things in those classes in obviously much greater depth. All right, we'll have a few of these little physiological concepts as we go through the course. We'll have one on the circulatory system, respiratory system, metabolism, and there's a couple more to try to tie together sort of important concepts in evolution as far as physiology goes. All right, so let's go on to our next animal group, which are the flatworms. So you would have seen these flatworms in lab last week. They're a pretty numerous group. Somewhere upwards of 20,000 species of flatworms. We can see in our phylogeny here, we've entered the Lophotrochozoa. So we finished off the two radially symmetrical groups, the radiata here, not shown as radiata, but could be written right here. We moved into the Bilateria, and we started on, this is actually the first of many protostomes that we're going to talk about. So we're essentially going to march down here. We're going to cover several more phyla today, a little rapid fire discussion of some of these smaller groups. The first one being the flatworms, or the Platyhelminthes. So here's some pretty pictures of pretty flatworms. Most of them are not pretty. I chose pictures of ones that are attractive because later in the lecture we're going to talk about ones that are not attractive at all. This group is our first group that's triploblastic. So up to this point we had sponges, which don't really have tissue layers. Then we had the Cnidaria and the ctenophores, both diploblastic, radially symmetrical. This is our first group that's triploblastic, got the mesoderm in there, and also the first group that's bilaterally symmetrical. So all the groups from this point on are going to be triploblastic, they're all going to be bilaterally symmetrical. So you don't have to memorize that with all the rest of the groups, just know that today, when you switch from the radiata to the flatworms, we switch to these new concepts, and then everything from this point forward are triploblastic, bilaterally symmetrical. Question?
Our test is on Friday, correct?
Your test is on Friday. Yes. And this will be on the. So here's the little Begizia, the one you looked at in lab, kind of cute. If you were to cut this animal in half and looked at it through its side, you would see that its body tissues are essentially wall-to-wall cells. There's no spaces or body cavities. So this group is actually one of the rare groups that we call an acoelomene, no body cavity. So it's a solid mass of tissue. The only gap here is the digestive tube that runs inside of the animal. So all three of these things are new. Triploblastic, bilaterally symmetrical, and it's an acoelomene. The The group contains those cute, free-living Dugesia that you saw in lab, but also a couple of less cute groups, the tapeworms and the flukes. The Dugesia are planarians. They live kind of everywhere. They live in marine habitats, they live in freshwater habitats. They can even live in damp soil, so in the terrestrial habitat. Most are microscopic, but this group has many species that can be absolutely huge. So you have some tapeworms that can be upwards of 20 meters in length. That's 60 feet long. So imagine one of those. Imagine the length of your digestive tract. You can have tapeworms in there that can be very many feet long, living inside. You may have guessed that they're all very flat. So their common name, the flatworms, comes from the idea that their body is flat. So they're not round, like you think of as most worms, they're flat. So they're called flat. So that Dugesia you looked at in lab, it looked kind of like this, flat. Tapeworms, flukes, flat. Makes them a little bit distinguishable from some of the other groups that we're going to talk about. Let's start with the cute ones that don't harm us. This is the planarians. Your textbook will also use the word hermilarians to describe this group. They're mostly free living, so they just do their own thing. They swim around or crawl around, not living inside of another animal. And most of them are marine, although we do have some species living in terrestrial and freshwater habitats. The one you looked at in lab last week, Dugesia, is actually a freshwater species of planarian. They have little cilia on their belly, which they can use to sort of creep along, and so they look like they're gliding from place to place. So if you watch them in the petri dish, you would have seen this sort of graceful ability for them to swim or move. You'll notice here in yellow, this is the nervous tissue. We've got a far more complex nervous system here than in any of the animals we've talked about so far. So the jellyfish and the cnidarians and the ctenophores have nervous tissue, which is, responsive to stimuli, but it's not exactly highly coordinated. In this group, and in most groups we're going to talk about going forward, you start to see clustering of nervous tissue in one part of the body into more complicated networks, essentially. So when you see a clustering of nervous tissue, a small clustering, sometimes that's called a ganglia. If you see a very large clustering of the nervous system, you might end up using the word brain. Okay, so there's like a difficult line to draw here. What's a ganglia, what's a brain, what's not neither. But that's the sort of idea we're talking about. So there's a clustering of nervous tissue at the front end of this animal. And you actually see some nervous tissue with the capacity for recognizing stimuli. So you would have saw little eye spots on your little Dugesia. That's not that uncommon in this group, okay? Especially the free swimming ones. And you can see nerve cords going down the rest of the body, connecting the rest of the cells of the body and helping with that communication idea. So they have a more specialized nervous system than the cnidaria, but as we go, we'll see even more complicated nervous systems evolving over time. One thing you may have noticed in the lab, in the manual, it talked about its mouth, and it asked you where its mouth might be. The mouth is actually kind of right in the middle of the body, underneath the belly. It's not where you would think it is, up at the front end. So even calling this a head is kind of challenging to do. It's got sort of clustering of nervous tissue at one end, but the mouth isn't even up there. It's sort of in. The other thing you may notice here is that there's only one opening to the digestive system. There's a mouth, but there's no anus. So we haven't actually got to an animal that has evolved a fully complete digestive tract away from that of the anus. So just one opening. So when this animal eats, it'll bring food in through the mouth, but when it needs to get rid of waste, it will actually eject that food back out the same. So pretty simplified body plan for sure. One of the reasons this group is incredibly interesting to scientists is because of their way of, one of their potential ways of reproducing. They can actually reproduce asexually through a process called fission, where the animal will just spontaneously carve off a part of itself, like pinch off a part of its body, leave it behind, and that part that's left behind will then reform into a new individual. So it's pretty neat. So you can see this little animal's going to turn into 3 parts, and this one's going to turn into 2 parts. Each part has the capacity to regenerate all the parts that it didn't originally have and reform an adult individual. So this is called fission, and it's seen in some animals out there, but not that many. And so obviously people who are interested in how organs regenerate or tissues regenerate are quite interested in this group. Imagine if you lost an arm, you could just grow a new one. That would be pretty useful. So people study this with the hope and aims that maybe we can find ways to regenerate human tissues in order to replace things that might be damaged or lost. You can actually take these animals and slice them into parts, and then each part will reform the missing parts and survive. Okay? So pretty impressive ability there to reproduce, essentially. They are hermaphrodites, so they can reproduce on their own asexually. They have both male and female parts, but they can also cross-fertilize. So 2 individuals who both have male and female parts can still cross-fertilize to create new offspring through sexual reproduction as well. So pretty cool group of animals. For the most part, friendly and not a problem. The other 2 groups, though, are big problems for a lot of people, and that also means it's important for us to understand. So the monogenians and trematodes are groups of flatworms that are collectively called flukes. So you might have heard of liver flukes or blood flukes. These are animals that cause different diseases in humans, but also in animals that humans care about, things like livestock. So they're parasites. So here's an example of a common life cycle for a lot of different flukes. A lot of times there's a human host, and these flukes are living somewhere in the body. And you can see male and female flukes here. They can reproduce sexually. They're usually attached to one another, and they're just constantly producing fertilized eggs. Okay? They're really good at that. And so what happens is the human host will shed these larvae into the water. So you can see a little ciliated larva here. And then contaminate the water. And then a secondary host will pick up that fluke larva. So a lot of times snails are the intermediate host, but some other invertebrates can be intermediate hosts for this group as well. And the larvae will grow and develop and change into a different kind of larvae in that host, and usually kill the host, and then be released back into the water, where if that person ingests some of the water or somehow it gets into their body, that will complete the life cycle, getting that larvae back into the final host, where it can then develop into an adult and then continue that tragic cycle. So this is meant to be a very generalized life cycle for the group, essentially. Some of these are very prevalent and very important medically. Something like 200 million people have a disease called schistosomiasis, which causes all kinds of terrible symptoms and death, potentially. And it's caused by the blood fluke schistosoma. So this is a group of flukes called schistosoma. So really common, obviously a huge problem, depending on what part of the world you live in. They're very stealthy, very sneaky. The worms have evolved to coat themselves with surface proteins called antigens, which mimic the host's body. So our immune system can't recognize them as foreign and then go and kill them off. They get in there and then they're able to stay there because we don't recognize and notice that they're there. And so for that reason, they can survive in a human host for long periods of time, continually producing offspring. They can then get released into the environment and potentially infecting more and more people. So although you may not care about flatworms, they are important from a human health perspective, because understanding their biology and their life cycle is obviously important for understanding how to control them or eliminate. All right, so those are the flukes, not to be outdone by the tapeworms, our another favorite group of flatworms. They're also flat. They have this weird body form. You can see these divisions in their body. They have these little divisions called proglottids, which we'll talk about in a second. I just want to point out one thing, so I don't forget later. We have a group of worms coming up called the segmented worms, where their body is segmented into little distinct sections. This looks like that, but it's not true segments. There are lines that look like segments, but they're not fully detached from one another. So we'll come back to that when we get to the segmented worms. This group looks like that, but it's technically not fully segmented. So the tapeworms, as you probably know, are parasitic. They live in usually vertebrates in the intestinal tract, obviously. And they're very clever. They're able to release these hook-like claws from their head, and they hook into the lining of the intestinal wall, and that holds them in place. So as your food's going through, they are not washed through with it. They stay in the intestine. And they just hang out there and literally feed off of your food and reproduce. They're really good at that. They don't even have a mouth. This group has, through evolution, lost its mouth because they just use their walls of their body to absorb the nutrients from the host. So all that food that you're digesting, they just sop it up, like right through their body walls. So very, very simplistic body, able to absorb nutrients from the host's intestine. Those proglottids, and there's many of them, are essentially their sex organs. They're pretty much just feeding, reproducing machines. Okay? They can release self-fertilized eggs. These ones are often hermaphroditic. They just produce their own self-fertilized eggs and release them into the digestive tract to be released into your feces and then out into wherever your feces goes. So you can imagine if your feces contaminates the water supply for another organism and they drink that or eat it, they're going to get these little fertilized eggs, which will then get absorbed into their body. And in a lot of cases, they find their way into the meat of animals we like to eat, which is unfortunate. So here are some encysted tapeworms in the muscle of cow and beef. And so when a person eats this meat, if they don't cook their meat well enough, the cysts, these dormant cells, will survive. They'll survive part of the digestion process, find their way back into the host's intestine, and then repeat that life cycle all over. So if you take anything from this lecture, cook your meat, become a vegetarian, whatever your choice is, because obviously you do not want this in your body. But obviously an important aspect of human health, if you're interested in human health, this is another reason this group is pretty important to understand. Anybody hungry? Okay, those are the flatworms. Let's move on to the next group. So most days we'll only cover one phyla, but today is a bunch of short ones. This one's only three slides. This is the rotifers, or Rotifera. It's a group you would have maybe seen in lab, but probably not, because all the rotifers died. So unfortunately, we didn't see live rotifers. About 1,800 species, really common in a lot of different environments. Here we are, little rotifers. Here's some pictures of them. You may have seen the video in lab if your TA put that on for you. Why are they important? Well, they're triploblastic, they're bilaterally symmetrical. We've heard that for the last group. What's new here is this is the first group of pseudocoelomates. So remember the the flatworms in general were acoelomate. The rotifers are our first group of the body cavity. Now it's not a true coelom as we would define it. It's a pseudocoelom, but it's a body cavity nonetheless. So remember, the pseudocoelom has a surface that's lined by endoderm and the other surface by mesoderm. The true coelomates, the entire body cavity is within a small difference there. Make sure you know that for the exam. So our first coelomates. They live in all kinds of different places, marine, freshwater, damp terrestrial environments, and for the most part, they're really, really tiny. We're talking microscopic sizes. So you need a microscope to view these. In addition to having the gastrovascular cavity that we've seen in other groups so far, like the jellyfish had one, the free-swimming Eugesia had one, this group is the first to have a full alimentary canal from mouth to anus. This is the first group where we see the anus essentially here. So we have a full digestive tract from front to back. Remember, digestive tract is often just called an alimentary canal. So we have a mouth and an anus and a tube that goes between. So here's the different parts. You would have looked at them in lab a little bit. Most of them are very see-through, so they're easy to see. I'm going to show you a YouTube video of them feeding, in case you missed that in lab. They're pretty interesting to watch. So here's a rotifer. I'm going to pop its head out here. There's going to be some other, like, I don't know, protist or something over here. So ignore that. Here's our rotifer. And the way they feed is they have this really impressive crown of cilia around their head. So you can see this, there's actually like two wheels that spin around. These little cilia are spinning around. And they create a water jet. The water around them will start to create a current that moves towards their mouth. And so any food particles that are in their mouth or in the water will go towards the mouth. The mouth will then pick them up and will swallow them to this apparatus called a trophide, which is essentially a grinding plate. These two things that can grind together and chew up the food. Not a true jaw as we would think of it, but it does the same kind of job. And then, as I mentioned, they're often very translucent, so you can actually see inside. You can see a lot of green in here. This one's probably eating some algae or some phytoplankton of some sort. But that's the generalized feeding mechanism by the road. So they're pseudocoelomates. They have a complete alimentary canal. That distinguishes them from all the groups that we've seen before. They also have one other rather strange way of reproducing that some scientists are quite interested in. In many of the species within this group, they're able to go through a cloning procedure where they can create clones of themselves asexually. Most species, most of the time, only exist as females, which is kind of interesting. And the females will produce unfertilized eggs, so no male involved or required, that then develop into new females. So imagine that society, no war, no famine, all that good stuff, right? Just females running the show. This process is parthenogenesis. So you'll probably have heard that term before. This is what it refers to. It's the production of offspring asexually without any requirement for fertilization. And it's really common in this group. Sometimes, it depends on the species, usually under unfavorable environmental conditions, a female will produce a male offspring, which will then be used for sexual reproduction with another female. So like I said, it's kind of rare, it doesn't happen very often, but it's usually when, for whatever reason, environmental conditions aren't very good. So here's a sort of typical life cycle of a rotifer. Most of the time, the usual cycle here is an adult female produces a diploid egg. So think about that, it's a diploid female adult. Usually when we talk about eggs that are haploid, they have half the genetic material. In this case, the adult female produces a diploid egg, so it has all both sets of chromosomes and all the stuff that you would require. And then that diploid egg will develop back into another adult female. Repeat that process almost all the time. Under certain stresses, this is the stress cycle. So imagine like cold temperatures, droughts, maybe there's limited food or some other issue that's going on. the female will produce, instead of a diploid egg, a haploid egg. So, pretty weird that it can switch to that. That haploid egg will produce a haploid adult male, which pretty much has no other role to play except fertilizing eggs. In fact, the males often don't even have digestive tracts or any of the necessary parts for feeding themselves. All they are is a way of reproducing with females. So they only have one purpose in life, to fertilize females. So they'll produce sperm that will fertilize a female haploid egg, or just a haploid egg, I should say, into a diploid, what we call resting egg. And so the resting egg is kind of like a seed, essentially, this egg that is able to stay in dormancy until conditions become more favorable again. So maybe it'll take days or weeks or longer, maybe months or longer, or years, before conditions become favorable again. And then that little diploid resting egg will hatch, develop into a female. And then the usual cycle will continue. So a very odd and different reproductive style, but that's something we see commonly in this group. Question here?
So would it be determined if it's going to be male, female, or just going to be man?
Which one, the last one?
The haploid,
This one here?
no.
So the egg would just be fertilized by the sperm, because it's only haploid, so it needs that other part of the genetic information. And then standard, normally it would produce a female that will then develop into an adult. Question in the back?
Is the diploid resting egg always a female?
Is it also?
Is it always a female?
I think that's essentially the question that was here. I think usually, yeah, because you need a haploid male to get to continue this cycle, and so it would have to go to the female and then produce another haploid male. Yep. Okay. Oh, question, yeah.
Have there been cases where they produce males that have been able to feed on their own?
No, they only have the one function essentially, and that's it. We will see a few other examples like that. Some females will figure out how to neutralize males that way. But it's pretty rare, obviously, in the animal kingdom to see this. But it makes you think, right? Think about if this is the normal cycle, probably everything you've ever been taught in biology is that sexual reproduction is good and necessary for the long-term survival of the species. You need crossing of genetic information or else bad things are bound to happen. Yet this group routinely does this. There's actually 360 species of this phyla, so not all of them, but 360, called the Deloidae or Deloid rotifers, where they, as far as we know, reproduce only by parthenogenesis. No one has ever seen a male in any of these species. Okay. So now we've probably only been looking for a couple hundred years, but still, no males to be found. By genetic analysis, it's predicted that these groups have been only reproducing by parthenogenesis for 35 million years. So, obviously this is possible and works in some situations. We don't normally think of it as normal, but it's definitely something that happens in the rotifers. So, another reason that rotifers are interesting is because of their pretty robust ability to reproduce asexually in this really weird way. So this is the rotifers. Unfortunately, there's a video here. It won't work. It's behind a paywall now. So it's a Science Friday video that's actually pretty good. So if you Google Science Friday and rotifer, Deloidae rotifers, you actually can find this video, but it won't work for me here, unfortunately. I've shown it every other year, so every once in a while, videos are. All right, so that's the rotifers. Any questions about that? All right, this is like rapid fire phyla introduction here. Go to the next group. This lecture actually contains 2 phyla, only 7 slides, so not too much information. But again, we'll highlight some of the special attributes or characteristics of these 2 groups. So this next group is called the Ectoprocta. This is probably the only day in your life where you'll ever think about Ectoprocta, but today's the day, so you gotta pay attention. Well, actually, you might think about it Friday too, depending on the file. This group is collectively known as the bryozoans. There's about 4,500 species of bryozoa. You've probably seen bryozoa if you've ever picked up a rock that's sitting in the ocean. If you see like this mat of what looks like algae growing off any rock, it's almost definitely bryozoans. There could be algae in there too, but the bryozoans are really common and they pretty much look like little algae or plants. So for a long time, they were mistaken for an algae or some sort of plant-like organism. But if you look closely at them with a microscope, you'll see characteristics that are much closer in resemblance to something like a coral than what a plant would show. So a really interesting group, very common, although there's not that many species, they're incredibly common in coastal areas. So mostly marine, there's a few freshwater species. They live in sessile communities. So if you remember, sessile means attached to something. So they're attached to a rock, or a boat, or a dock, or something. They've got like a sort of gritty, tough exoskeleton. So they feel like a plant almost. Kind of rough. These animals are suspension feeders. And what suspension feeder generally means, and you'll see different definitions, is they're not actively doing much to feed, but they're putting out an apparatus that's able to collect food. So it's not that different from filter feeding. I don't like those two terms because they're hard to differentiate. So these animals put out this fan of cilia. You can see it here. Here's some other pictures. And food items and particles will get stuck to it and then get transferred into the mouth, which is at the base of the little ring of cilia. This ring of cilia in this feeding structure is called a lophophore. So that's a term we introduced a few lectures back. So it's one of the defining features of the Lophotrochozoa, this feeding lophophore. So the lophophore is a crown of ciliated tentacles that surround the mouth, and they sort of extend through that sort of rough exoskeleton, and it's put out in the water for things to catch onto. Food goes into the mouth, and notice a complete digestive system. So a tube that's running from mouth to anus, kind of like the rotifer. That was the first time we saw this, now we see it again. And that's going to become the norm. Most animals have that from this point on. There's a few exceptions, but most have a complete alimentary system. What we haven't seen yet is this is the first animal group that has a true coelom. So they're coelomates. The rotifers were pseudocoelomates, and now we have the evolution of a true coelom, body cavity fully lined by mesoderm. So these are obviously some of the characteristics that make this group special, and why we have to talk about them in this kind of lecture. But they're weird in other ways too. So this group really challenges your definition of what an individual is from a colony. Kind of like when we talked about corals, each polyp I said should be thought of as an individual, but they live in these large colonies, creating larger structures. This group, it's even harder to define, because each of them can change their form into different forms. So let me explain. When you see these bryozoans, often you'll see them looking like this. There's some sort of stalk that's attached to something, and then you'll have branches of different parts. Most of them look like this with that lophophore, that feeding apparatus, but not all of them. Other ones may look different. It turns out that this group is made up of parts which we call zooids. So it's like animal-like parts, if you will. And each zooid modifies to perform different roles for the colony. So there are feeding zooids, called autozooids, that do what we talked about before. They have the lophophore, they can feed, and they can absorb nutrients, and then they can pass those nutrients on to the colony. But then there's other zooids that modify to create different parts. They'll create like the attachment stalk. or they'll create different defensive spines that can help defend the colony. So if something tries to take a bite out of it, they get a mouthful of this hard, spiky material. So those are called kinozooids. And then some of the zooids actually modify into different reproductive zooids too. So you'll see ovizoids, where is it? Ovicells, sorry, that produce eggs. And then you'll also find testes, which are a different zooid, if you will. They can produce sperm. So on a colony, you'll have both male and female parts. They're hermaphroditic. They'll produce eggs and sperm. The eggs and sperm can be released, so they can cross-fertilize with other bryozoans. But the overall idea of what's an individual versus what's a colony is pretty gray. It's hard to really differentiate. So you can think about each zooid as a part of the whole animal, but it is hard to imagine where you draw the line between individual and colony. So those are the bryozoans. I promise the next lecture will be all on one group, but today we've got lots of little groups as we go. Any questions on bryozoans? You should be noticing me focusing on certain things like, hey, this is the first time we saw a coelom. Hey, this is the first time we saw a full elementary canal. This is an evolutionary change through the tree of life where those things now become sort of the norm as we go. So when you're studying, keep those things in mind for these exams. Our final group for the day are the Brachiopoda. This is another one of those groups where we don't have that much to talk about, but what we have to talk about is pretty important. Okay, so we're coming down the list here. Brachiopoda, only about 300 species. Why would we bother spending any time on this? There's over 1 million animal species and we're talking about 300. If you look at these, they look like little clams. Okay. They're actually collectively called lamp shells. That's their common name. And although there's not that many species today, historically, this is one of the most abundant animals on the planet. So if you look at the fossil record, lots and lots of lamp shells. For whatever reason, they're going extinct, and there's only a few species left. But even though there's only a few species, there's still lots of individuals out there. Most of them are pretty small, so here's a fossil with a lamp shell. You can see it's smaller than a penny. Most of them are sort of these little tiny guys. That really do look like clams. And we'll talk about that similarity in a second. So why are they important? Also triploblastic, also bilaterally symmetrical like everything else at this point forward. We also have coelomate here, okay? So like the Ectoprocta, we have a coelomate here. All of them are marine, and like I mentioned, they're often mistaken for bivalve mollusks like the clam. So you would have saw the clam in lab last week. Spent a lot of time getting up close and personal with the clam. If I gave you one of these, you would say, well, that's a small clam, because it really does look like a clam. There's no doubt about it. But let me try to point out a couple of distinguishing characteristics. The first is they have something called a pedicel. So this is not something a clam has. Clams just bury themselves in the sand. They don't have an attachment. The pedicel is like a little stalk that these lamp shells will attach to the surface with. Okay, so a little pedicel that anchors them to the substrate. So that makes them different. As I mentioned, they were very abundant back in the Paleozoic and Mesozoic eras. And if you look inside, you'll see that they have a very large feeding apparatus called a lophophore. So that lophophore we saw in the last group, also very common here. And this is how this little lamp shell works. Food enters the shell, gets stuck on the lophophore, this ciliated sort of structure, and they get passed to the mouth of the animal. Now, hopefully what you're thinking is that looks a lot like the gill of the clam, which it does. It has a lot of physical resemblance to the clam's gills, but it's a different structure of lophophore. So these guys have the lophophore, the bivalve mollusks do. And that's going to be our next topic on Wednesday, the bivalve mollusks. So to close out this group, the last thing I'm gonna talk about, the last concept, it's gonna challenge your three-dimensional interpretation of what this animal is. And I'm sorry, but I don't have a better way to explain this, you just have to take my word for it. If you compare a lamp shell, the brachiopod here, to a bivalve mollusk like a clam, there's one last difference. It's almost impossible to know, but I'm gonna tell you about it anyway. Their symmetry, or their line of symmetry, so how you would cut them in half, is technically 90 degrees to each other. Now, like I said, you would never notice this unless someone pointed it out. It's really hard to even explain with a two-dimensional picture. If I were to hand you one of each, you would see that one opens, this is my best impression, like this, and one opens like this. Okay? So, not a big difference. But if you're trying to distinguish them, a little clam versus a little lamp shell, that would help. So their line of symmetry is perpendicular to one another. So here's a brachiopod opening this way, and here's a clam, or so this is actually a mussel opening this way. Okay. The worst pictures I could show, this is the best pictures I could find, but it doesn't really convey any information. Even if you find a textbook that tries to explain this is what you get. Okay? You have a picture of a lamp shell over there, the brachiopod, and a bivalve mollusk over here. The line of symmetry is this way in the lamp shell, so you can see left and right are the same, but then it opens like that. Whereas the line of symmetry here is down the shell, essentially, so both sides of the shell are symmetric. So small difference, but technically a difference. So we'll mention that as a defining characteristic of this group. Next class, we're going to talk about bivalve mollusks. It will be our last lecture before the midterm. And on Wednesday, I'll tell you more about the midterm. So we're out of time. I'll see you on Wednesday.